Hello students, welcome to the second session of Principles of Inheritance and Variation. In the previous session, you had learned about law of dominance. It states that when two alternative forms of a trait or character are present in an organism, only one factor expresses itself in F1 progeny and is called dominant, while the other that remains hidden is called recessive. When experiments on peas were repeated using other traits in other plants, it was found that sometimes the F1 had a phenotype that did not resemble either of the two parents and was in between the two. The inheritance of flower color in the dog flower or snapdragon is a good example to understand incomplete dominance. In a cross between true breeding red flowered and true breeding white flowered plants, the F1 was pink. When the F1 was self-pollinated, the F2 resulted in the following ratio: 1 red is to 2 pink is to 1 white. Here the genotypic ratios were exactly as we would expect in any Mendelian monohybrid cross, but the phenotypic ratios had changed from 3:1 dominant is to recessive ratio. What happened was that capital R was not completely dominant over small r, and this made it possible to distinguish capital R small r as pink from capital R which is red and small r which is white. What exactly is dominance? Why are some alleles dominant and some recessive? To tackle these questions, we must understand what a gene does. Every gene, as you know by now, contains the information to express a particular trait. In a diploid organism, there are two copies of each gene, that is, as a pair of alleles. Now, these two alleles need not always be identical as in a heterozygote. One of them may be different due to some changes that it has undergone, modifies the information that particular allele contains. Let's take an example, a gene that contains the information for producing an enzyme. Now, there are two copies of this gene, the two allelic forms. Let's assume that the normal allele produces the normal enzyme that is needed for the transformation of a substrate S. Theoretically, the modified allele could be responsible for the production of the normal or less efficient enzyme, or a non-functional enzyme, or no enzyme at all. In the first case, the modified allele is equivalent to unmodified allele, that is, it will produce the same phenotype or trait, that is, result in the transformation of substrate S. Such equivalent allele pairs are very common. But if the allele produces a non-functional enzyme or no enzyme, the phenotype may be affected. The phenotype or trait will only be dependent on the functioning of unmodified allele. The unmodified allele, which represent the original phenotype, is the dominant allele, and the modified allele is generally the recessive allele. Here, in the example above, the recessive trait is seen due to non-functional enzyme or because no enzyme is produced. Till now, we were discussing crosses where F1 resembled either of the two parents or was in between. But in the case of codominance, F1 generation resembles both parents. A good example is different types of red blood cells that determine ABO blood grouping in human beings. ABO blood groups are controlled by the gene I. The plasma membrane of the red blood cells has sugar polymers that protrude from its surface, and the kind of sugar is controlled by the gene. The gene I has three alleles, IA, IB, and IO or small I. The alleles IA and IB produce a slightly different form of the sugar, while IO does not produce any sugar. Because humans are diploid organisms, each person possesses any two of the three I gene alleles. IA and IB are completely dominant over IO. In other words, when IA and IO are present, only IA expresses because IO does not produce any sugar. And when IB and IO are present, IB expresses. When IA and IB are present together, they both expresses their own types of sugars. This is because of codominance. Hence, red blood cells have both A and B type of sugars. Since there are three different alleles, there are six different combinations of these three alleles that are possible, a total of six different genotypes of the human ABO blood types. ABO blood grouping is an example of multiple alleles. Here, you can see more than two alleles governing the same character. Since in an individual, only two alleles can be present, multiple alleles can be found only when population studies are made. Occasionally, a single gene product may produce more than one effect. For example, starch synthesis in pea seeds is controlled by one gene. It has two alleles, capital B and small b. Starch is synthesized effectively by capital B homozygotes, and therefore large starch grains are produced. In contrast, small b homozygotes have less efficiency in starch synthesis and produce smaller starch grains. After maturation of the seeds, capital B seeds are round and small b seeds are wrinkled. Heterozygotes produce round seeds, and so capital B seems to be the dominant allele. But the starch grains produced are of intermediate size in capital B small b. So, if starch grain size is considered as a phenotype, then from this angle, the alleles show incomplete dominance. Therefore, dominance is not an autonomous feature of a gene or the product that it has the information for. It depends as much on the gene product and the production of a particular phenotype from this product as it does on the particular phenotype that we choose to examine, in case more than one phenotype is influenced by the same gene. In this session, you have learned about incomplete dominance, which is a phenomenon in which F1 hybrid exhibits characters intermediate of the parental genes. You have also learned codominance, which is a biological phenomenon in which alleles are able to express themselves independently even when present together as codominant alleles. With this, we come to the end of today's session. Thank you and have a nice time.